So today we don't have a whole lot on the agenda. We've got two exercises from the end of last class that we're looking at doing stuff with ModelDB, making sure we were comfortable loading some models, running some models, and so forth, modifying the models. Before we do that, though, does anybody have any other questions or comments that they want to raise? All right. Yes, sir.
I have it. Yeah, thank you so much actually for the great lectures so far. I have a general question about the working the already uploaded models in the NeuronDB. So does it make a difference that whether the model made in the any specific operating system, like in the OS or Mac, or like in Linux? Since I'm just as an example, I downloaded one model, not the one in the exercise that like in the other one, and it sounds that it just works. It only works on the Linux system, not on the Windows or even not on the Mac. And in the Windows gives me one error, in the Mac another error, but in the Linux it works perfectly.
So I guess the short answer to that is that it's entirely possible to it's entirely possible to do things. If you're using the stuff that we're teaching you in this course, everything should be, all of the neuron stuff should work across the board. But it's possible to do things that are going to break that. So for example, in Python, you can have commands that send stuff to the shell, right? So like if you say, by the way, here's a shell command for making a directory to put the results in. And if you write that in a way that's OS specific, then obviously it's not going to work in a different one. But there are also OS general ways of doing this, right? Python has an OS thing. The other main gotcha where you see this compatibility issue is that some of the mod files, especially for older models, have what's known as a verbatim block. And a verbatim block is an opportunity to, instead of using the sort of semi-structured definition of a mod file, to actually just go in and put arbitrary code in. And so people write C code. You don't see this that much anymore because Python is so powerful. But in the old days, there was a lot of arbitrary C code in mod files sometimes. And
so by old day, we're talking about how long ago? Since the model that I'm working on is for 2011.
Yeah,
that's far enough.
People reuse mod files that they find because it was in a paper that they liked, and the paper did a model that they liked. So for example, just yesterday, I was working with some students on a model of epileptogenesis in dentate gyrus, the one by Santa Kumar as the first author. And it had about a half a dozen mod files that gave the Mac users among the students a real hell of a time. It worked fine for the Windows and Linux users, and it all tracked down to many of the all these problematical mod files had procedural blocks in them, just procedures, not functions, not things that should return a value. But for some reason known only to the people who wrote these mod files in the first place, they put in a verbatim block that says verbatim, return 0, end verbatim. Okay? Arguable that they shouldn't have bothered doing that, that it shouldn't break anything, but it gives the Clang compiler that under Mac OS is now being used. That simply rejected the file, wouldn't compile any of these 6 mod files, and gave an error message about a procedure returning a value. So yeah, and this is a paper that was done published in 2004. It produces nice results. It produces results that are understandable. It's been cited a lot of times. It's still being cited now. Anybody who goes back and gets that and is a Mac user is not going to know why it doesn't work for them. So I was just going to ask Robert today, do we need to now start going and prophylactically purging verbatim blocks from ModelDB entries? And maybe the answer is as we discover them. Okay, so that's what that's the kind of thing can happen.
So I'll say this. So one of the other things, just because something was done in 2011 doesn't mean it was originally done in 2011, right? So they may have been using something from who knows how long ago, and just reuse. And that doesn't mean that it was the right. It doesn't mean that it was the right way to do things at that time either. So sometimes people just use what they find works, and which is good. But if you don't really understand what's going on, this is why we're really trying to stress you understand, then you end up with some bit of hacks that are maybe not always ideal. And I have some specific examples in mind. If you find yourself thinking that you need a verbatim block, I would argue that you don't, and talk to us.
Yeah. For example, Bill Litton has a bunch of really nice models that are in ModelDB, and his lab publishes prolifically. But they have taken advantage of verbatim blocks to implement all kinds of stuff. And that all kinds of stuff is kind of opaque, and I haven't been on an occasion yet where I've seen it not work, but I sure wouldn't want to have to reuse any of that code in my own stuff, because I wouldn't know what to do with it, and I certainly wouldn't know how to debug it.
Like in continuity of my question, in general, not in a specific like an example, is there any possibility that we can fix the errors across the platform and make it like an platform independent thing?
So in general, I mean, in theory, basically, right, all of these programs. So to answer your question, the literal way that you ask, yes, of course, because all of these things should, Mac, Linux, and Windows can do all of these things together. If, on the other hand, what you mean by we, so there's a couple of questions as to what do you mean by we. And so Ted suggested, for example, as things come up, we can try to address them. It's just so that most everything on, not some of the newer stuff, unfortunately, but almost everything in ModelDB has an associated GitHub entry. That would be a place to raise an issue, a pull request, if there was something. It's just as you find things where there is a compatibility issue, I'd say if you can fix it yourself, that's great. If you can't, might be good to ask us and see where we are.
So is it okay I share like an error that I received with you?
Oh, that's great. Yeah, that's terrific. Would you please?
Sure, I definitely do that. Yeah.
Yeah, and through the NEURON forum in particular. But there is a real problem. We cannot reach into all of the hard drives on all the computers around the planet where code is lying around. And old code that gets reused that had something that worked long ago when it didn't matter, and may have worked as recently as last year, may be superseded, may now become not merely deprecated but obsolete and broken because of updates. And there's no way that we can possibly make everything compatible with all past code. If you want to see an example of what you do when you try to make everything you're doing compatible with all past code, take a look at Windows. Microsoft Windows has attempted that for decades. It has never actually worked properly. They have never been completely compatible with all past code. And now if you look at the updates and bug fixes are being discovered every month on a routine basis, many of them just trace back to old garbage that is still in the kernel that you're using. Okay? So backward compatibility and making everything robust are not compatible. Utopia is not here. It will never be here. That's not the way people work.
There's another obscure little gotcha that I'm actually trying to track down and have not, but so one of the things that's sort of underlying this, so IEEE came out with a standard in, I don't know, maybe the 1980s for how floating point arithmetic works. And so the deal is, if you have addition and subtraction and multiplication and division, you ought to get the same result regardless of your platform, because the standard says everything needs to be accurate to within what's known as one machine epsilon, or the smallest possible difference between two floating point numbers. Unfortunately, a lot of other functions that show up in neuroscience mathematical equations are allowed to be up to 2 machine epsilon away from the truth. And so then now, so now that you have this wiggle room, you can get more than one answer that's consistent with the standard. You do have pathological cases wherein you may actually see subtle differences between different platforms. If all of a sudden if your model is so sensitive that that matters, then that's a problem with the model more than anything else. Like, but I've definitely seen things where, so this is what's known as sensitivity analysis, and there are tools like Uncertain Pi that allow you to look at this. I have seen, so it's not that hard to find things where you may stumble across one of these places where a different platform will give you something that's like 15 bits out of a difference. So, you know, the relative error is extremely small, but non-zero. And I have one pathological example on my screen right now where I remember Linux window. I think Linux and Mac disagree on what a certain number raised to the 4th power is, which is fun. Again, many bits out. The last possible bit out. They differ, but.
So even if we tidied up NEURON completely, we couldn't possibly tidy up things that occur because we're reusing libraries that are developed and maintained by others.
So, but this should be mostly an issue of quantitative identity versus anything else. If you're seeing qualitative differences, that's a problem with the model more than anything else.
Which is why somebody once wrote an article not that long ago, it was in the last decade, I'm sure, about what should we be striving for in terms of precision in models of biological phenomena. And should we be concerned about exact quantitative results, or should we be concerned about qualitative results? And the end conclusion that the authors came up with was qualitative results. Of course, that begs the question of how sloppy is too sloppy. And boy, I just don't have a good answer for that. On the other hand, if you spent some time in a wet lab, actually doing experiments on real animals and being involved in experimental protocols, especially behavioral experiments, you realize that there are so many potential sources of error coming with the data that we reuse in our computational models as the basis for our models, that maybe we should be a little forgiving of our own numerical results and accept some slop. Anyway.
So I just shared a link in the chat here to this paper that came out last year entitled Non-Replicability Circumstances in a Neural Network Model with Hodgkin-Huxley type neurons, wherein they do run into this machine epsilon error issue, and they do see some divergence between in the exact code between different operating systems. And so this was not neuron code, this was C code, but just throwing that out there. So they see the burstiness, they see similar looking shapes, but the bursts happen very decidedly at different time points. And I don't know a lot about this paper, so that's really all I can tell you, other than that it exists.
It looks like a system in which the state space has regions of rapid motion and regions of slow motion. And it looks to me like the problems are emerging in areas where there's slow motion, because that's where driving forces may be small. The net driving force for motion in any direction may be small, but that's because it's a small difference between two large numbers. And that may be the perfect setup for the kind of circumstance where problems arise. If you go back to the figure that compared those different results, you'll see here that the one that is, there are a couple that are extremely problematical, and this the middle set of traces has a very long, I'm going to call these black things when they go up, I'm going to call those plateaus or bursts. And you'll see that there's a very long interburst interval during which the derivative of whatever the y-axis variable is noisy and averages about 0. And there is this other state variable whose time course is in red, and you'll see that this red line comes asymptotically toward a some kind of a quote threshold. And that is also highly problematical, because when you get that close, the difference between, again, where you are and where the steady state is becomes very small, a difference between two large numbers, and there round off errors, all kinds of numerical precision issues come into play. And it seems like that's what's going on here. A robust simulation, a robust time course would probably be one in which the red line, the red traces never get that flat with time, and even more robust if the black traces had more slope and not simply were not simply governed by noise. It looks like noise is playing some role in this. It's always possible to concoct something that exposes problems with numerical precision, and I think the authors, being clever people, have managed to do that. I'm not too surprised that there are differences. Anyway, you had something of substance you wanted to talk about, other than the general moans and groans about numerical methods, right, Robert?
I did, but I'm kind of curious if Ben wants to elaborate on this and on his moan and groan about numerical methods.
I was just trying to call to find the talk. I'm not on a great connection right now. But I just remember the math department. I don't recall if they were using the time step or an error tolerance. But they were just using that as a bifurcation parameter, and I thought that might be a cute example, but I can't find it right now.
I mean, so again, I don't want to overemphasize this either, because what we're talking about is things that sort of pathological cases that can happen. That doesn't mean that you would expect them to happen. All right. Any other questions or comments? All right, so hearing none. So last time we ended with two sets of exercise. We talked about ModelDB, we talked about model sharing, we talked about a little bit about our own various experiences trying to reproduce models and what worked and what didn't work. So we ended up with two exercises. So this first one here said to download and examine the model from ModelDB, Model 2488. Some simple basic questions, who are the authors, what questions were they studying, asked us to use ModelDB's model view tool, how many different models would you say that are available, and then look at one specific one, see if we can run it, and then see if we can change it. In particular, what we want to change is to drive the cell in a different way. So did anybody do this? Does anybody want to? I'd love if somebody else wants to talk through their take on this problem. All right. Well, that's okay. Let's go ahead and try it anyway. So if I take this, I open up ModelDB 2488, which is a classic model by Manin and Sinowski that I think we've brought up. I know we've brought up on various occasions as an example. In particular, so they're making the point that if we know the ion channels, that doesn't mean we know how the cell is going to behave. There's more than just the genetic information to describe the response properties, or at least more than just the channel expression information. We have to consider, for example, also the morphology. All right, so who are the authors? So this is obviously Zach Manin and Terry Sinowski. This is the paper, right? Influence of dendritic structure on firing pattern in model neocortical neurons. We can go to PubMed, look it up. From there, we can look it up on Nature's website or wherever. We can also see, just out of curiosity, if we check the citations tab, there have been a number of models that are in ModelDB that cite this particular paper, and some of them are quite recent. So here's a 2018 model where in the underlying work, the paper cites this model from 1996 of Manin and Sinowski, 2009 paper, 2007, 2009, 2014, and so on. And there's a whole bunch, actually. I'm still scrolling. Look at this list. There's a whole bunch of papers. So the models that we have the code for are, pulled out with the bullet point in bold. But there's an awful lot of modeling work that's somehow the corresponding paper cites back this 1996 paper. So that's interesting. So let's look at the model itself, right? So we said that they're interested from the model information. We have a description. So they're looking at 4 reconstructed neocortical neurons. So they've got layer 3A spiny cell, layer 4 stellate, layer 3 and layer 5 pyramidal neurons with active dendrites. Let me just make this a little bigger. Running the simulation demonstrates that an entire spectrum of firing patterns can be reproduced in a set of model neurons, which share a common distribution of ion channels and differ only in their dendritic geometry. So that's nice. So we've got information, we've got a description, we know who wrote it, we know why they wrote it, and we've even seen what kind of an influence that this has had on the field, at least as judged by the citations tab. There's an awful lot of papers that have then gone on to cite this work. So let's look at model views and try to get a sense of what all it is there. So we see that we have 5 different views that we can look at. So we've got this one for each of these cells, and then there's also one that's, I think, figure 2. So I can pull up one of them. All right. And this, actually, zoom this out a little bit. This was figure 1A, a layer 3A spiny cell. What do you make of this morphology? Let's look at this as compared to, let's say, a layer 3 pyramidal cell. So they look very different, right, in terms of appearance. It's a layer 5 pyramidal cell. So one of the things I'm just sort of throwing out, one of the things that they looked at, for example, is they did some things with very stylized geometries. They're like, let's say, so this layer 3A spiny, it's just sticks, right? It's just a whole bunch of sticks. And this is not a reconstruction. That's fine. But so they looked at this at a variety of morphologies. We can see the different morphologies. We can look at the cells. Oops, we can zoom out way too much. Yikes. We can zoom out so much we lose everything. I'm just going to reload this page because it's so bad. All right, so let's go back. So this is the Fig 1C. So we've got 4 fundamentally different cell types, and let's look at figure 2. And figure 2 we see is in fact just literally one stick. So you could argue that there's 5 different models that they looked at. Oh, apparently it's 2 sections, sorry. So we've got 5 different models that they looked at with their channel distributions. And then I asked specific questions about figure 1C. These are some questions that might give us some sense about how they did the numerics. So how many sections are involved in this? Clearly, this is a realistic model that's come from a tracing somewhere. So let's look at it. So there's a total of 117 sections and 257 segments. We can look at, there's 12 inserted mechanisms. It's a variety of ion channels. There's calcium channels. There's calcium gated potassium channels, sodium and potassium. And then we can always go and say, well, hey, look, what is this sodium channel that's involved? Well, that's inconvenient.
Actually, it looks fine in our computer, so we can download the files.
Okay, well, okay, so it just doesn't like me. That's fine. That's kind of the best outcome, right? So yeah, so we can always go back to the model file tab. We can always look these things up. Here's the KM. Yeah, I know exactly why it didn't work. But so here's the KM. It's the potassium channel, this muscarinic. And then we've got a whole nice little description. There's a warning about temperature. Remember, there's a whole discussion about that. We can see the parameters that are the defaults anyways. We can see the equations. We can see how they wrote things. Awesome. Okay. So we've gotten some sort of a basic sense. We see the cell. We know what's kind of there. We know what channels are there. I can see that, well, the potassium conductance is uniform. That channel is uniform. This channel, well, is mostly expressed in one location, but only has 2 different values. Sorry, I've zoomed this in and I can't see anything. Yikes. Sorry. Let's just ignore that window. But so we can see that these things are fairly uniform. Okay, we get a sense, we can explore what's there. Here's sodium, cool. Sodium has 2 different values. We've got this value of 20 over most of the cell, the sodium conductance. And then there's this one little place near the initial segment where the sodium conductance is much, much higher. Cool. All right, so we've gotten a sense of this. So then, well, okay, we've looked at this a little bit. How do I actually run this though? Anybody remember to figure out how to run one of these guys? How do I actually run this model?
In the references, it's mentioned.
Yeah, let's look under the references. So references, run protocol. And this is actually the part that at some point, this has been, since this was generated, the folder path has changed. And I don't know how or why that would have happened, but so this is no longer accurate and this needs to be regenerated. But in any case, once I'm actually running this, I get some instructions. I'm going to load this file called mossinet.hoc. You'll find that for a lot of files, that's used as an entry point. I can select the general figure. So in this case, figure 1, I can do figure 1C and then run it. So let's see if it works. So this is what it says. So I'm going to download the zip file. And what it's done is it's created this folder called cells. And then I will open my terminal. I said I will open my terminal. And I'll make this bigger so we can see it. All right. I'll go into the downloads folder, this folder called cells. Okay, so here's everything. I will compile my mod files, enter an IV modal. It runs, it gives me a bunch of messages as it does it, but doesn't take so long. Now I want to run it. It says to use this with NEURON GUI - Python. Personally, this is usually, I don't want to say this. This is usually the same thing as just using Python. But since it says to use NEURON GUI - Python, I'll go ahead and do that. And the one difference that you do have is it pops up a NEURON main menu. And it gives you some extra text as it starts up, like, you know, what version you're running. All right, so then I say from NEURON, import h, GUI. I'm just going to follow the instructions that it said here. I uploaded, oh, crud. I updated my machine right before doing this. Never ever do that. Huh. That's going to be a problem. I'm going to propose a 5-minute recess. Wherein I try to reinstall Anaconda so it's not angry at me. Let me try this one other way before I do this. Python none. Is it going to be angry too? No, that's okay. So we're going to run it this way because my computer's angry at me. But this will work. So instead, I will just follow the same instructions, but from inside of Python. From NEURON, import h and GUI. At that point, this is the difference. It pops up the NEURON main menu. h.load file. I'm just going to follow the instructions. Mosanet.hoke. I'm going to learn how to type. There we go. h.fig_1. It's going to set us up for running the figure. It's going to say h.fig1c. Is apparently doing something. And h.run. I'm going to be very cranky in just a moment. Yeah. Where are my windows? They're there. You see them, right? You see how it says, oh, look, all those windows are here. Where are my windows? Okay, so one of the, nope, I just disappeared that window. Shoot. They are hiding. Let's try this one last time. I do apologize for this. I think what I need to do is disconnect some of these monitors. Boom, from NEURON, import h and GUI. There it is. h.load file, mosanet.hoke. There it is. h.fig_1. All right, now this is when my windows disappeared, but there is a figure one. So let's see if we can find it. So one thing that you can do in NEURON is you can pull up what's known as the print and file window manager. And so this is going to give you a sense of this whole big square here, a big rectangle is my screen. And I can use this and I can select move and I can move a window around. So you'll see I'm moving around this select a figure window. So my other 3 windows that are hidden are over in another monitor. But I would just drag them over, but it's not being cooperative, so I'm going to move them over this way. So there's my graph. There's my run control. And there's my choice of cell things. That's very strange. So you'll notice that this is very wide. I have 3 monitors running, and I'm sharing a specific one, and it's not seeming to play nice with Zoom. So in any case, so if I say h.fig1c, I've selected my model, and then I'll just say, oh wait, here's another one. It's popped up now. There's fig1c, here's my cell, and I can run it, and there we go. So I see this bursting behavior. Okay, was a bit of an adventure to get here, but we did get a bursting behavior. We did get figure 1c, and if I had disconnected my other monitors, this would have gone much smoother because everything would have appeared in this window. But you got to see the print and file window manager, and you learn how you can move things around. Yay. Okay, so if ever any window doesn't appear, that's a place you can look to see where they are and just move them around. All right, so in any case, so here's the question. So we ran our cell, we got some action potentials going on, we successfully selected the cell that we wanted. And I could, let's say, if I wanted to look at what this looks like, if I wanted to look at the membrane potential, I could go into plot what, select the membrane potential. Oh, wait, sorry, and I'll say shape plot. There we go. So right now the cell is at rest basically everywhere. But if I were to run this again, just for kicks, well, we wouldn't see anything apparently. It's all happening so fast. Never mind. All right, well, okay, so this was a bit of an adventure, and I apologize for that. But in any case, we got our cell to run. The instructions worked. So that's good. Any questions on this so far? Okay, so then my next task on this was, why is this cell firing? So did anybody look at this? Did anybody figure out what it is that makes this cell fire?
There's
And
a
how-
current source.
There's a current clamp. How did you know that?
Tools, point process, viewer.
All right, so I can go to tools, point process, point manager, I guess, or viewer. Would you use?
Viewer.
And sure enough, there's a current clamp at the center of the soma. There's another way that we could have figured this out, incidentally. Back on ModelDB, we have this model view. We say, hey, look, there's 1 point processes of 1 base clamp. And in particular, it says that there's 1 current clamp. And here are the properties for that. And I see this, you see this big dot at the soma? Yeah, that's a current clamp. That's at the center of the soma. And here are its properties. And you'll notice that those properties there match the properties that a point process viewer tells us. So that's always a good sign that everybody agrees. So starting at time 5 milliseconds, nothing happens until 5 milliseconds, and staying and running for a time of 900 milliseconds, which is why you see it kind of shutting off. We're going to inject 0.1 nanoamps of current.
And since you were able to use that ability to expose the object, a graphical object that manages that current clamp, now you can easily do interactive exploratory simulations. You can change the duration, you can change the magnitude of that current clamp. You'll have an instant reminder of what the parameter now is. If you don't like the fact that the clamp starts so early, you can change the delay, make it start later, verify that the model has properly initialized to steady state before it got stimulated. So you can see that you have exposure of key aspects of the code through ModelDB's introspection, that model view that it provides, and you also have the ability to explore interactively through a graphical interface. So you have a lot of power to work with these models.
Yeah, and you can mix and match. And I don't know if we've talked about this, but so you'll see that I've been adjusting. So that was at 0.4. This is back to 0.1, but here I started a little bit later. What did I want to point out? So you'll notice, if you haven't noticed this already, anytime you change something, you'll get a little checkbox here, and that check will be that box will be checked to indicate that it's not the default value. You can uncheck it to go back to the default value, and recheck it to go back to wherever you left it the last time it was checked. You can also right-click on this, on the checkbox, to give you the option of changing the default value, making it now default as to 0.5. And then now if I go back to 0.1, now 0.1 is a change, and we can go back and toggle back. So you can always be like, you just sort of have your baseline, do your experiments, and you'd be like, wait, now what did I actually change? Well, you can actually see, because anything that you've changed from the defaults has that big old red check on it. Also, if you didn't know this, if you right click on the up and down arrows, you can change how it is that you're, what an up arrow and a down arrow means. All right. The other thing that I'll point out, so we've seen this, we've done some experiments. I'll also point out here, you'll notice that this is current clamp 0, h.i clamp square bracket 0. So let's go back over here. If I say, well, it doesn't say h dot, it just says I clamp. So if I say h.i clamp square bracket 0, hey, look, it's a thing. It's a thing that has a duration. Oh, the duration is 900. Well, what if I change the duration over here in the terminal to be 100? Well, you'll see that immediately the graphical interface saw that it changed, gives you this little check mark to indicate that it's not at the default, but is in fact at 100. So we knew what to call it because we knew what it was in the point process viewer. So it said iClamp 0. So this is h.i clamp 0. And incidentally, what is this? If I give this a variable name, just because I'm having a moment here, and then I can do the completion. So if I want to know where it is, it's just IC. So if I've stored my current clamp in the variable IC, then I can say, well, where is it? Where is it? It's IC.getSegment. It sets soma of 0.5. This, by the way, is a segment. So it has a section property and an X property, and you can use that for whatever you want. If you need to know, wait, where is something again? So that's nice. Yeah.
That's a very useful trick, by the way. Creating an alias, a Python alias for a thing that you only know the object name of. It's always very fragile to use object names. It's always better to create an alias for it and do exactly right like Robert showed here.
And so just to give an example, so imagine, for example, that you've saved a session and you're like, okay, well, everything is, everything's consistent. But all of a sudden now you've created another iClamp before you've loaded a section. Then all of your iClamp indices will be off by 1 because these things just get numbered in the order that they're created, right? So if you use names are consistent. If you use numbers that assume that everything was done in a particular order, you risk running into problems. Okay, right. So let's say that I have my current clamp, it's IC here, and I want to just turn it off. So I changed this amplitude to 0. Sure enough, in the point process viewer, the amplitude 0. That's not surprising. If I go and run the simulation, not a lot happens. Cool. All right, so we have now done the experiment where we're able to remove the existing stimulus. So then in our exercises, we were asked to say, well, okay, so disable that which causes the cell to spike, we use the point process viewer, or we use a model viewer. We've done a number of things. We found out what it was. We've turned it off. And now it says, can we drive the cell with the Poisson process spike train? And then using Python, record and plot the soma's membrane potential as a function of time. Well, let's do the first part first, which is the Poisson process spike train. So how do I get a spike train in NEURON?
Using netstem.
All right. So I'm going to create a netstem. What about the netstem? What are my options and parameters?
Start.
Yeah, so if I can actually see this by, if I sort it to a variable, I can just do in as dot and then hit tab, I think twice, and then it'll show me what all of my options are. So there's a start. All right, that sounds promising. So let's start this at some time, I don't know, 50. Let's say that I want a number of these things. So give me maybe 10 stimuli. And then what's my interval? My interval will be, I'll have a spike, give me an input every 50 milliseconds. And you know what? I said that we should do this with a Poisson process spike train, but let's not jump there, because if I introduce randomness immediately, it's going to be sort of an extra little bit of a confound, and I don't know what it's actually doing. So let me start out with a very deterministic thing, so I know exactly when this is going to spike. All right, so I've created my netstem, but I haven't hooked it up to my cell. So I need to do two things to hook that netstem up to my cell.
We need a synapse and then a netcon.
Yeah, I'm going to need a synapse and a netcon. So I can just go ahead and create a synapse. Let's just say centered at a synapse position it somewhere, and I'll use an EXP syn just because it's built in the NEURON and we're familiar with it now. Nope, sorry, it was actually h.soma. Going to do synapse at the center of the soma, and that that synapse has certain properties as well. It's got a tau, a time constant. So we could have this be relatively fast, relatively slow. I don't know what time constant makes people happy. Doesn't matter, 3 milliseconds. All right, that synapse has a time constant. What other properties does a synapse have?
Reversal potential.
A reversal potential, that's the E. So let's say that I want this to be an excitatory synapse. So I'll set it to 0 millivolts, let's say. And that's really what I need here for that synapse. So now I have a place, I have a spike train, which I could think of as basically, there's some cell that I haven't modeled explicitly, but there's some cell somewhere that's going to give me some events. I have a synapse that's listening to them, and now I need a netcon to connect the one to the other. And so I just go ahead and create my netcon, so h.netcon, and that's going to be my netstem, which I called ns, and I'll just connect it up to the synapse. And remember that netcon, very importantly, has some sort of a weight. We don't know what it needs to be, so let's just pick a number. It has a delay. If we're running a parallel simulation, for going from one node to another, we need a non-zero, we need a positive delay. But since we're not doing all that parallel infrastructure, and since we're really sort of driving from the outside, let's just go ahead and make that delay 0. So now I have my synapse, I have a netcon that's connecting it to a source. Let's see what happens if I just hit emit and run. Well, that's kind of fun, isn't it? Does it make sense? Well, so there you go. So what's your take on this? Does it make sense? What are obvious things to look for here? We said there'd be an interval of, we said that it would start at time 50. Okay, that's, I see that going up around time 54. I don't know if you can read the numbers on the screen there, but that's what it says. And this one's at 105, and this one's at 156, and this one's a little over 200. And this is a little after 253. This is peaking a little after 300, a little after 354. So it does seem like we're getting this this input happening every 50. Why? What do you make of this fact that it's we only get one burst? Why do you think that is?
The netcon weight is pretty low, so the initial events are sub-threshold.
But yeah,
And- And then owing to integration after like around the fourth event, we get a spike maybe.
yeah, I agree. So let's look at this. If I zoom in here, well, it's not really making any clearer. But you may be able to notice, let me make this a little bigger here. You may be able to notice it's not going back down quite to the baseline. It is inching up ever so slightly with each time. So let's see, let's just suppose that the net stem had more of them. Do we think, it's a hypothesis, that if it inches up ever a little bit after each time, then maybe if I run it enough, then I will see it's firing another burst. Let's find out. Let's an end run. We got that burst. We're still inching up, but we never see it. I could run this longer.
I think what I would do myself as an experimentalist is I would apply a brief excitatory input at just one. And I would make that be a current clamp. And I would like to see the trajectory of membrane potential after the burst. I'd be looking at post-burst hyperpolarization, which I think without synaptic inputs, because I think that's what's actually governing the interburst interval here.
Oh, now that I agree with also, but I did want to just sort of point out that we are getting this buildup.
Oh, absolutely. Now, it could be also interesting to make that be a stochastic input.
Yes, so let's do that. So remember, to make it be a Poisson process, all we have to do is set the noise flag to true. Let's go back and not run this quite as long so we can actually see what's happening. So just run this to a time 1,000. I'll end and run. And now we don't have to actually wait so long. It's actually kind of funny. It seems to fire faster with the noisy inputs. Which if you think about it, makes sense, because you could have some that go together and give you that build up faster than you might want.
So you get a cluster of excitatory inputs, which very rapidly makes it fire a burst or two. And then you have these sparse areas where there aren't very many inputs, allowing the mechanism to recover from the most recent burst.
And then it still fires. You know, that burst is still more likely to happen sooner. So let's go back and compare. So that was with noise. And then if I flip it to be without noise and I run it, that noise makes a very real qualitative difference in the burstiness of it all.
So here you got 4 bursts, when the previous run you just got 1 with totally deterministic input.
Mm-hmm. I'm not sure if you call.
Natural inputs tend to be a bit noisy, by the way.
So that's fun.
Yeah, the only time they might not be naturally noisy is, say, if you go to a rave and you just listen to the drum. But aren't raves supposed to be sort of numbing?
So I guess the one takeaway here that I want to highlight is this introducing this. it's not just that you saw this, it's not the case that we see the same major behavior except at slightly varying time points. We're actually seeing things that are somewhat different just because we have that noise that we'd never see if we didn't. And here we get things more than, more extra spikes because there's another input at the right time. So yeah, and obviously we could do the same exact thing using the GUI entirely. So we can take tools, point processes, managers. I can do a
Well, yes and no. You can create a point process manager for the NetStim, and you can create a point process manager for the XPSIN, but when you connect them, you can only do that with code. And it's a very cheap way to build a graphical user interface that contains NetStims and XPSINs to use the GUI to make the graphical interfaces for those tools, but ultimately you have to have a bit of code to make the netcon.
yeah, there is a network builder, but that doesn't quite do the same thing here.
Yeah,
I don't think that's going to work for us.
It's cumbersome. You can do it. In some ways, it's convenient because you don't have to write code. It's a very good toy tool, toy prototyping tool. But I think it's better to combine code and selective use of individual GUI tools rather than to use, that's like the network builder tool is kind of like a giant Swiss Army pocket knife that's got 100 different blades and weighs 5 pounds.
All right. So yeah, the other thing that I wanted to sort of highlight, by the way, so now I've been doing all of these experiments within this interactive terminal. But what I'd really like to do, you could imagine, is I want to go back and I want to get back to this. I want to have a script that's going to get me back to this situation where I've made my model. And there's a few ways that we can do this. Does anybody have a favorite way of going from the terminal to a script? All right, well, let's do it this way. This is this way we don't have to remember anything. So what we can do, so one thing that I could do is I could just Google for Python show history. And so Python is smart enough to keep track of all of your command line history. So it's all there. If you're using IPython or Jupyter, there's very easy ways of doing this. If you're not, you have to use this readline module. Stack Overflow is a wonderful place to get this and to get answers to questions. Oh, so there we go. It did it already. So this is my readline history. This goes back a good ways, but in particular, the stuff that I care about started with this from NEURON import H GUI. So I'll just take this, I'll copy that, and I will open up a new text editor. Sorry, I said I will open up a new text editor. Here we are. I will paste that in. This is everything that I've done, but in particular, there's a lot of the setup, figure 1C, blah, blah, blah. We did grab the current clamp. We do want that. We don't need this part where we did the experiments of grabbing the sections, but we do want the part where we turn it off. We do want the part where we set up the NetStim, set up the synapse. That was me making a mistake. Here's the netcon creation. Give me a bunch of inputs. and then do it with noise. So if I have this, then in theory, if I save this, let's just save this somewhere, and I will call this, well, this is 2488.py. Let's call it my 2488.py because we changed things here. Right, so this, oh, that needs to actually be in the same folder as everything. That's not going to work. But I'm going to copy that to this folder. Nope, that's not where it is. No, I'm just going to, okay, I'm just going to save it into that folder, which is the easier way of doing this. So downloads, cells, my 2488.py. So let's go ahead and I will say that I want to run this now. So I want to run this interactively. I don't want it to shut down when I've done it. So python-i my 2488.py. All of the windows appeared on the other screen. So what I'm going to do is I'm just going to switch my screen that I'm sharing rather than try to drag them around again. So here they are. All right. So now everything should be set up. So now if I say and then run, there we are. We've got that noisy stimulus going into the center of the soma. It's not quite as, on the one hand, it's not quite as clean. It's not quite as simple as just being able to save a session, but we were able to get the entire session history out pretty straightforwardly, I think. And there we go. And I had suggested to record with Python, and actually I wasn't going to do it, but on second thought, I think I am. So if I were to just modify this, just to show you how little there is to this, I want to record time. Okay, so I'm going to create a new vector, and I'm going to record h.ref time. I want to record membrane potential, so I'll create a new vector, and I'll record h.soma of 0.5, let's say ref v. And then now if I f initialize this guy, f initialize to negative 65 millivolts, and I do this continue run to time 1000 milliseconds, and then
minus 70.
Minus,
model is initialized to minus 70.
Yeah, so it is.
Well, you could have debugged that, but you know.
Well, it would be different. It wouldn't necessarily be. It would have done something different. Yeah, yeah. So this model is initialized to minus 70, so I can plot this T and V, and I can show that. And I didn't import the plot, so import matplotlib.pyplot as PLT. Boom. Save this guy. And I'll quit it, and now I don't need this to be interactive, so I'll just run what we did, and we're running the simulation. We're getting the pretty pictures, and it showed up in the other screen, but there we go. The other nice thing is the matplotlib window does in fact match what we did. It was supposed to, but nonetheless, the point is this model, we didn't really dive into it. It's not even written in Python. There's literally not a line of Python involved in this model specification, but that didn't hamstring us. That didn't really affect us. We were able to use the tools, figure out what things were called, read stuff in Python, plot stuff in Python, do whatever we want with Python.
Including discover what was in the model itself. Okay, so now you had an idea of what biology is represented in this model and how you were able to work with it, and you did not have to be a Hoek parser. Okay, so you were liberated from that.
And so Hoek, for those who don't know, is another programming language that NEURON supports. Since this is a Hoek model, we are going to actually go back in on Thursday and talk a little bit about what exactly is Hoek, how do we make sense of it, and more importantly, where are the things that if we do look at some Hoek, I wouldn't I wouldn't ever expect you to write Hoke. There's no reason in this day and age to write Hoke. But if you do come across this thing, like we did with this particular model, can we make some sense of it? Can we, figure out what's going on? And so I call my lecture for Thursday, Hoke for Reading Knowledge. Okay, so I think that pretty well, yeah, sorry, go ahead, Ted. Ted, were you saying something?
No, you're fine. You're good. You're good. You
Okay.
go ahead and say exactly what I was thinking. Go ahead.
All right, anybody have any other questions or comments? All right. So hearing none. So our second exercise was to look at a different model. So this is the model for ModelDB 239421. So here we go. This is model entry 239421. Who are the authors? So what are the basic things? Well, Mazzoli, D'Angelo, okay. Synaptic activation of a detailed Purkinje cell model predicts voltage-dependent control of burst pause responses in active dendrites. That's certainly a descriptive title. There's a nice little write-up here. This is, I believe this is actually an excerpt from their abstract indicating their study and how they looked at it. So a couple of notes on this one. Unfortunately, this model, for whatever reason, does not have a model view available on ModelDB. That's sad, but that's okay. We're not going to let that stop us. All right, but I can see at least what kinds of conceptual questions they're looking at. This one should be, it looks like we should expect some more bursting in this. Well, let's find out. So who are the authors? What question were they studying? What ion channels are present? Well, we know there's a potassium channel. We know there's a calcium-gated potassium channel. If you're wondering about what any of these mean, you can always just click on them. So it's IKCA here. And this is going to give us an example. We're not discriminating within ModelDB right now. BK versus SK. But fundamentally, anything where we have a potassium current that's activated by increases in intracellular calcium concentration, they're being lumped in as this IKCA. We can go back, we can look at the model files if we want to see what else is going on. We see that there's a mod files folder. I can open up the mod files. And in fact, there are several different mod files. There's several different mechanisms. You can click on one of these. And so here, for example, is a NAV 1.6 channel. And this is their kinetics for that. So this is a model that's described by a kinetic scheme. I don't know if we've actually seen one of these things, but it's basically a forward rate and a backward rate. We go from one state to another. It's an alternative way of expressing model kinetics as opposed to just writing down the equations. And you can develop some really sophisticated, really complicated models this way. And I mean, so not everybody does this, but we do have some sort of a description of all of these things. We can see if I look at this NAV 1.6 channel, this exact model has been used in 3 other models. Of course, they were all by the same first author, but it does at least give us, and they're all in the context of the cerebellum. But it gives us some things to think about as to if we're trying to decide if we want to reuse it. All right. So that was the ion channels that are present. If I just go back here, the next question I asked you was, where are the parameters specified? Did anybody have any luck with tracking this down? And if so, how did you come across this? How did you decide? Well, there's two sort of obvious places. Well, there's three things that I think are pretty obvious to look. Anybody have any suggestions as to what the obvious places to look are? All right, so we've got these various model view param subset.txt file is the first suggestion on here. Well, there's a bunch of numbers, not super helpful. They're all integers, which is a little suspicious. It was a reasonable guess. There's another one, again, sort of mysterious numbers. That's not helpful. I do have this PC param thing though. That's the only other place where there's this param. Oh, you notice that's not even param. That's, I don't know what PARM is, but in any case, so here it looks, we don't know, but it certainly looks like we're setting a bunch of parameters here. Now we don't know where these are being used. We don't know if these are being used. We don't know if things are changing after this fact, but certainly at least we've explored and we've thought about, we've clicked on the files, and there are certainly things that look like they could be parameters. So what we can do is that we can go ahead and run it and see what happens. And see if these are the numbers that we see when we look at various places.
This style of programming is meant to make it easier to understand models.
Yeah, it's done a nice job. This is one of the things that I do really like about this is that it's putting all of the stuff that's doing one kind of thing together, right? Like here's where the parameters are. You know, you could also imagine, and here's where the topology is, and here's where the ion channels are put in. You know, if you can build things in a modular way, that's always nice. You might see, taking this a step further, you might see things like a JSON file or some other sort of config file would be another way of sort of generalizing this instead of doing this in Python. But I think this makes it pretty clear. All of the parameters are here. If we want to change something, we'll change it in one file. We can track the changes in that one file only. And we don't have to go digging around to figure out, wait, where was the value for ELEAK set? All right. So in any case, let's see if this is going to work. And this is actually possibly an interesting question, given that this computer seemed mildly unhappy after my last, when I tried to follow the instructions the last time. All right, so here are the files. There's a folder called mod files. There's a folder called protocols. We see them. There's a whole bunch of instructions. We should read the README. Should always read the README. Usually it's intended to be read. So here's their instructions, blah, blah, blah. This is done with Python with NEURON. It was actually originally done with 2.7 and 7.5, but they checked that it works with Python 3.6 and 7.8 as well. They checked a couple of other versions. Awesome. All right. So it's got some instructions as to how to set this up. So in particular, inside the Purkinje cell folder, we should run this inner NIV modal ./mod files. And so that's saying Sorry, that's not what that's, what was this file called? Purkinje cell with a capital P, okay. Purkinje cell with capital P. It says that I should run NEURON model ./mod files. Which is saying I didn't put the mod files in this folder, I put them somewhere else. Namely, all of the mod files are in one location. You'll see that some people do that. Some people leave them in the main folder. It depends on whatever you're comfortable with. I think they're both reasonable ways of doing it. But it's always a thing that if there's any doubt, if they're not just in the main folder, if they're put somewhere else, it's always good to tell somebody where they are and how to compile. So NEURON modal ./mod files, it says in the README. And then it says models provided with one protocols are able to reproduce parallel fibers, ascending action, stellate cell bursts, run NEURON GUI, blah, blah, blah. And then this is the one that I'm worried about, wherein we see if it's going to be angry at me because of updating the Mac without testing this. Yeah, that same error again. It works on my home computer. This is because something's, actually, let me try this real quick. I'm gonna just pip install neuron pip install -- force neuron. Let's just see if I reinstall neuron, if that'll magically fix everything. Look at that. I reinstalled neuron. Also, installing neuron is easy and fun. Okay, so anyway, so I reinstalled neuron.
Great, now we have a Mac that acts like a machine running Windows. That's the usual Windows fix. Well, if you can't use Word, reinstall Word.
No, to be fair, I updated to the latest version. I updated the operating system yesterday, and I had not tested to see if that. And I think some compiler library changed. So.
Likely story.
Yeah. Anyways, at least it was an easy fix, right?
Even if we don't know how it worked.
It's like turning it off and on again. All right, but so in any case, so I followed the instructions. After reinstalling Windows, I get this bursting behavior. I get this firing behavior. It sort of matches. Here you'll see this is run for a longer period of time, but we get a similar output. All right, yikes. It's still running going forward. You can see that it's still running. If I wanted to stop the execution, I could always stop it. There's a view equals plot, which we're still going, but this is going to go to time 5 seconds. So I'm just going to stop it because that's a while. But there you go. So I can view equals plot, and this is what we actually computed. Okay. So we got our input. The cell fired. That's great. We reproduced more or less. You know, we ran it for a different period of time, but we reproduced the figure that we expected to see. That's good. We noticed, for example, well, let's just see. It says that there's protocols /CO1. Are there other protocols? No. But we can look at this. We can see some things about it. So in particular, they're explicitly running with fixed step. Yeah, I don't want to dive in it too much, but in any case, so we can see this is the thing that we're told to run. We can look at it. We can see we've got a lot of parameters in here. So what was our exercise? Oh, I did want to point out one other thing. Sorry, I do want to run this again. So if I run this again, and it's running its calculation, and that's all exciting. But let's go over here and let's just open up a new terminal here. So you'll notice that NEURON IV, which is our main process here for NEURON, is using 900% of the CPU, or so it's varying. So this is an example, and one of the reasons that I wanted to highlight this particular model is this is an example of a different type of parallelism that we haven't really talked about. So we talked about using parallelism with MPI. This is, however, an example of parallelism using threads. So NEURON can take a model and split it up using threads, and you basically, if you're using the GUI, there's actually a little checkbox that can be that simple. If you're doing this programmatically, you basically specify points where you want the model to be broken up, and that's pretty much it. You can check out the code for this to see how they're doing it. But so this way, we're able to take advantage of having a mini core system. NEURON's not running single threaded. NEURON doesn't have to run single threaded anyways. And so this is an example of that. So that's cool. And if you didn't know about top, you'll notice that I stopped it, and now it's NEURON IV modal. No, sorry, now NEURON IV is no longer up here, and Zoom is using the bulk of my CPU. All right, and you can do something similar in Windows. All right, so let's get rid of that. So I did have some questions about this model. Oh, okay. So one of the things, one sort of downside of the multi-threaded case is if we do flip up multi-threaded, it does cause some problems with model view. So there was a question though, how many sections are in this model? Well, how are we going to answer that if we don't have model view? I mean, it's clearly, if it's using 9 processors worth and it's still taking a noticeable amount of time, like there must be a lot of complexity here. How do I find out? What are my general sets of tools here? I can imagine lots of ways, things I could do.
You could just do a for loop for section and all in h.allsec for seg and sec I plus equals 1.
Yeah. So let's do the just yikes. Okay. Sorry, this is getting, why is this hating me? I can't even delete today. That's how the day is going. Sure, so I could say seg count is equal to 0 for sec and h.allsec. For seg and sec, we don't actually need to, we can simplify this, but just for seg count plus equals 1. Okay, and how many segments do we come up with? 1,611. By the way, just for the record, if I want to simplify this, if I want to do something simpler, I could say, what's the length? If I were to make a list of all of my sections, so list of h.allsec, and then I could just ask for its length. So then there's, I notice the number of sections is equal to the number of segments. Is that right? Let's find out. In order for that to be true, every section would have to have inseg of 1. So let's just look at what is sec.inseg for sec and h.allsec. So this is create a set of all of the possible values of inseg. 1. So literally every section has exactly 1 segment. So there's 1,611 sections, 1,611 segments. I could also look at a shape plot. Here's the cell. there's a lot of complexity, and then there is a relatively simple, what I assume is an axon maybe coming off of it. But this is my my Purkinje cell. All right. So we've taken it, we've loaded it, we've done some analysis. One thing I said, so then, okay, so what happens if I double the somatic leak? So, well, one way that I could do it, we said we thought, here was our experiment anyways. It was back under Purkinje cell. Nope, that, yes, go away, stop. In Purkinje cell, there is this PC param. And we have this thing that says conductances for all of the channels with the same order as in the template. Okay, fine. So in particular, I have a soma leak. And so I could just double the soma leak if I wanted, let's say. And then, so my question is, well, how does that affect the behavior? Well, I can just rerun it. Up arrow, by the way, in my terminal. And can anybody tell me how this is different from before? It's pretty obvious, right? There's a significant delay before we get any action potentials, as Salvador commented in the chat. And then what we do get is we just see that tight burst. We don't see any of the other behavior. So now we've gone from something that fired, maybe a little burst sprinkled in there, to something that really only does the burst. So somewhere I would argue that that's a qualitative change. We could try to figure out where that qualitative change happened, and how would you go about trying to track down where that qualitative change happened? Does the question make sense? How do we go? So before, it was firing constantly, basically, once it got started. Let's just go back. It's always good when you do these experiments and you find a change of behavior to make sure that you undo the change that you think you made. Make sure that you get the old behavior back. Otherwise, you made a change that you didn't know. So remember, nothing's going to happen until I think time 200, but it won't take too long to get there. So here, this is firing fairly steadily until maybe around 600 or whenever it is that it does that really dense burst. So what's the solution? How do we go back? Like, I don't even mean in a programming way. How do we find where this behavior changes? I see somebody unmuted. Oh no, I guess they're back. Yeah, so there's a suggestion on the chat, which I agree 100% with. So do a binary search. So we know, what's that mean? So that means we're going to take this. We know we have one behavior at 1 times the value, and we have one behavior at 2 times the value. And then, so what we can do is we can do a binary search, and we assume, for the sake of this, we don't know this to be true, that it's not constantly switching between these 2 behaviors. But so let's just say, you know, if I go, instead of 2 times, what if it was 1.5 times? I could go do this experiment and run it. And then presumably, unless there's some other qualitative behavior in there, and there might be, not saying there's not, presumably it's going to be either this thing where it's continuously firing, or this thing where it's waiting and it's really only during these bursts. Well, at 1.5, it seems that we're in the same regime as we were in with 2, where we're waiting, and then around 600 here. There we go. There's a nice little tight burst. And we're going to go chill out again. All right. So I can stop this. I can get out of it. I can say, well, okay. So if 1.5 and 2 do the same thing, well, what about 1.25? And then I can run 1.25. And then I can just repeatedly narrow down until I see, you know, however narrow of a band I want to have to see something. It seems like we know what this one's going to do. And you start to get a little angsty. You do want to make it, you know, so then you could go 1.125, right? Just divide the difference. So we knew it was between 1 and 1.25. Well, we'll just divide that difference in half again, go back, rerun it. And then we were able to narrow down the window and narrow down the window and narrow down the window until such time as we've decided that our window is narrow enough. And so here, for example, with what is this? An extra, multiplying it by 1.125, we're in the first case. So now we know that the behavior is switching. We assume, we're making the assumption that the behavior is not like switching randomly in between. We don't know that. We don't know that there's not some complicated relationship. We have not proved that in any means. But if that's the case, that it does go from one to the other behavior, that switch point happens between 1.125 times the initial value and 1.25 times the initial value. And each time we can narrow down by a factor of 2 until we can decide with whatever accuracy that we want as to where we think the transition happens. Does that make sense? And if you're sufficiently ambitious, you can go ahead and automate these binary searches, but we don't need to here. And it would, I don't want to say this. I love, you know, it's fun to write programming problems. It's fun to solve programming problems. But if you're actually doing something wherein you can do like 10 experiments in the space of a couple of minutes to answer your question, maybe do the experiments instead of trying to automate it. Like, you know, there's a lot to be said for just trying things and seeing what happens. All right, so that's really all that I had that I wanted to talk about. Does anybody have any questions or comments about how we address this? How, you know, about ModelDB, reusing models, or really anything else that we've talked about throughout this class?
Well, I would highly suggest downloading models from GitHub, not from ModelDB, because Git allows you to search easier, and git grep is my favorite command. And if you're downloading many models and you want to make good decisions which model to choose to reuse, it's much easier with Git. This has been my experience for the last couple of years.
Yeah, and so just to highlight, so almost everything, and we really should make it everything, but almost everything is available through Git. So github.com/modeldb repository, and then the model accession number as well. So I can just clone that, and then that gives me a folder called 2488, and there's everything. And now I can do a git log, let's say, So we see that 2488, having been around since 96, has actually had a few changes that have been made to it over time to try and make sure that it continues to play along with some nice features, which kind of ties into some of our earlier discussions about, well, things that may have been right at one time or may have been good enough could be done better. So for example, the thread safe issue, typos, some buttons were added, support for better, more appropriate numerical methods, etc. So if you use Git, you get to see the history as well. Yes, sir.
I have a good question. Can you show us in this particular model how we can view the neuron morphologically, like in the view the model?
I'm sorry, can you say the question again?
Yeah, the question is that how I can view the like in the it says that they are using cerebellum Purkinje GABA
Yes.
cells. Can I view the model? How can I see the model?
Yeah, so let's go back to this. So if I go back to this Purkinje cell, and we had, this was how we were running it, right? So if I were to go and I were to run this, and it starts out just kind of doing its thing. So I'll just stop it because I don't care. I don't actually need to run the simulation. But what I can do is I can always just go to graph shape plot. There's my cell. Does that answer your question? Or?
Yes, yes.
Okay, cool. Actually, let me just show you one other thing for the record. Yeah, so you can go to, if you go to GitHub/ModelDB repository, you know, so we have the models here, we have the model histories. Most of them are pretty much, don't, you know, whatever information you can go in, you can click on one of these guys. It has the link back to ModelDB. Looks like, okay, so here we go. I click this at random. But this is an old model, right? This is, you'll notice that normal ModelDB models have, recent ModelDB models have 6-digit repository IDs. This is a 5-digit repository ID. But there was a question that came up back in February, I guess, of, well, how do I run this particular model, which is quite old. Apparently, judging by this text, looks like the model goes back to 2004, right? How do I run this model using what's known as core neuron, which is a part of neuron that allows the use of GPU simulations. It's a part of neuron that uses some support, it doesn't support all of the models. There's some limitations as to what it does, but for what it does support, it runs them in general faster and using less memory. Well, but this one, this particular, somebody asked us, how do we get this particular model to run with core neuron? Because it was unclear how to do it. So, okay. So one of our friends actually at the Blue Brain Project, which has done a lot of development work for core neuron, came back and said, well, okay, well, if you do want to run this model, this 17-year-old model predates any of these numerical solvers, here's how you do it. And so I mean, so this is not changing the code, this is not changing what it does, this is still a faithful reproduction of this, but now it's, at least we're able to deal with it and to use more of neuron features that we couldn't use with it before. So again, if something does come up and you have an issue, how do I get this one to work? Go ahead, open the corresponding issue, and make a commit. All right, anything else that anybody wants to talk about today?
I hope this isn't an annoying question, when you mentioned temperature dependence in the first model,
Yes.
I wasn't sure if it was a trick question because that model is designed to operate at only one temperature, but if you change the temperature, it will change the model.
Is that true? Oh, yes, it is true. So yeah, so in particular, that first model here, there's a thing, it's ran at a certain temperature, 37 degrees Celsius, but there are in fact 5 mod files that reference Celsius. I don't think did I ask about temperature?
Yes, it's in the slides.
Oh, okay, yeah. Oh, is it temperature dependent? So I guess it's a kind of, right? I guess, yeah. Sorry, that was not intended to be a trick question. What I wanted you to notice was that these mod files use temperature. And so if I were to look at them, let's just look at them here. You know, Celsius, where is this actually used? They calculate this, they use Q10 and this whole Q10 business and adjust some of the scaling accordingly. So this is a mod file that depends on temperature, but you are quite correct. The comment says, really, we're just talking about 37. We make no claims about other temperatures. So yeah, sorry, that wasn't intended to be a nasty thing. It was just intended to notice that some of the mod files use temperature. All right. Anything else that anybody wants to bring up? All right, well, if not, just to wrap this up, today is Tuesday the 27th. So we have 2 more sessions left in this webinar series. On Thursday, we'll talk a little bit more about HOKE. We'll have some exercises for that. Ted, are you gonna, do you have, I know you and I talked about a thing. Do you want to give a spoilers or? All right, well, if not, there's some other things that'll come up as well, but we'll see you on Thursday. Thanks, everybody.